my pleasure to introduce Dr. Anna Gaglianese, and she got her master degree in biomedical engineering and a PhD in neuroscience from University of Pisa. And then she moved to Netherlands, pursued her postdoc researcher in a 70 group at University Medical Center of Utrecht. And then she come here and she got a Curie Fellowship, working on investigate the functional architect of the multisensory motion system using 70 MR and transcranial magnetic stimulation. Her research focus is combining high-resolution imaging of cortical networks in vivo with advanced modeling techniques to probe our understanding of visual and multisensory processing in both health and disease. So thank you very much, and the floor is yours.
So thanks, Beijing, and thanks, CIBM, for inviting me. And thanks for being here for you, for the one that are here. So I have to introduce you something about I feel the fMRI, and how we can use it to study brain processes. So a bit about me, as Beijing said, I'm a biomedical engineering with a PhD in neuroscience. I always have a strong interest for the brain and how to study the brain, and I always use MRI to investigate brain function. So I always be involved in all the setup, starting from the protocol design, setup, and sequence optimization for the particular question that we have, to more data analysis. So trying to use modeling to explain data, and all of that always to explain some fundamental function in the brain. So fundamental visual neuroscience and multisensory function in the brain. So why MRI? So we can see the brain starting from really genes and neurons, so the microscale, going through more a microscale of networks and brain system, and the final goal is always to understand behavior or mental disorder. And I've always been fascinated by MRI because it really allows us to investigate the brain in really fine details. So thanks to structural MRI, so quantitative MRI, we now have a parcellization of the brain region. And thanks to fMRI instead, we can also give a function to the different area, the different structural area, and have this beautiful image of visual cortex, for instance. And I started with a 1.5 Tesla during my PhD, ending up with a 70 in my last year. And so during this changing between the field, one of the main questions is always why we do need the 7 Tesla. So why we need to go ultra high field? Well, the main reason is that increasing B0, we have an increase in the net magnetization, and this allows us to have more signals. So if you extract a ball of 7T or 3T, and you extract the signal to noise ratio, you have this huge increase in the signal available. So you can, having more signal, you can go higher in spatial resolution, and also really important in fMRI, you can decrease the number of trials that you need to reach the same significance that you will reach, for instance, with the 3T. So this is one of the main things why the magnet are increased in the field. And another reason is that with standard 3 Tesla fMRI, we can reach usually a 3 millimeters voxel in the human brain, while with the 7 Tesla we can really reach the from 1.5 millimeters to laminar fMRI, that is around 0.700 micrometers, and we can be really more specific to neuronal activity. So we can really try to remove the draining veins in the first millimeter of the cortex and be more specific. And different techniques have been now developed to achieve this really high spatial resolution. It's always a trade-off. So if you choose to go more spatial resolution, usually we lost temporal resolution. But there have been a lot also of new work done on the time resolved fMRI, so trying to go really sub-second scale in fMRI using different acceleration technique. And this is also quite important because, well, neuronal activity is in the scale of milliseconds, and we are measuring something in seconds. So we really need to go fast to understand better what we are seeing. But this is now really possible with the 7 Tesla. So new and new sequences and methods were developed to achieve this. And what I always find quite amazing is that with 7 Tesla was possible for the first time to probe in vivo neurophysiology in humans, in vivo, like non-invasively. So for the first time, it was possible to have ocular dominance maps of visual cortex, and also orientation maps. So these are work from Japan first, and this is from Minnesota. The 7 Tesla scanner, and was possible to have these beautiful maps for the first time in vivo in humans. And this was possible also because 7 Tesla really increased the specificity that we have with the neuronal activity. And for that, while in my time in Utrecht, we developed some modeling to see how close we were with bold activity and neuronal activity. So in Utrecht, they have this the epileptic unit there, and there are patients implanted with the electrocorticography grid. These are these ones. So these are implanted on the cortex, and they stay like that for like 1 week. And we had the opportunity to make some measurement while they are implanted. And basically, we did the same experiment in the fMRI scanner, and after the implantation with the grid. It was a standard visual stimulation, so standard dots like moving. So we extracted the neuronal activity in the high frequency band for the CHUV electrode with the convolve with the standard hemodynamic response function, and what we obtain is basically an estimated CHUV ECOG-based HRF. And we did the same with the fMRI. So in this case, we have the fMRI signal. We do the deconvolution because it was a really fast TR, and we have a measured HRF, and we find a strong linearity between the two measurements. And also, not going into detail, but also at the different feature of the visual stimulation that we were looking at. This was even more striking for the high density CHUV combined with the 7 Tesla fMRI. So these are high density grids you can see here. So this was the same. So basically, the high frequency band signal was extracted underneath each electrode, and also they went through fMRI before. This was a finger tapping task, and the bold signal underneath each electrode was extracted. And we developed these two. Basically, we compared these two spatial profiles, and you can see that are really strikingly similar. So what we see with the really neuronal activity and the bold 70 is matching completely. So this was quite important results to start to use more 7 Tesla fMRI really to probe neurophysiology and to look into the brain, the human brain in vivo, completely in vivo. And this is a node. We also developed together with Mariana Branco in Utrecht a post-processing pipeline to co-register this patient went through a CT scan, an MRI, an fMRI scan, and we developed a pipeline to co-register all these three together and have really a co-localization of the grid on top of the fMRI. This was done also with the 7T, and it's also used by neurosurgeons to guide surgery in epileptic patients during implantation. So now I want to show two examples, so different studies that I performed that using different 7T here in the CIBM facility, and also in Spinoza in Amsterdam and Utrecht. Showing two different points of view, so how we can use 7 Tesla for different purpose. So the first study was using a 7 Tesla fMRI to explain fundamental visual nerve processes in humans. So trying to go from monkey studies and rats and trying to explain neuronal computation in visual cortex for different visual stimuli. And in this case, we wanted to achieve high spatial resolution because we wanted really to get into to define topographic maps of neuronal ensemble, and also going into high temporal resolution. So because we really wanted to model the data. So this is the setup that we use at the 7 Tesla in the UMC. We reach 1.2 millimeters isotropic in the brain and 800 milliseconds temporal resolution. And to do that, we use this surface coil. So basically, these are coils that are put inside the head coil in the 7T and were located in the area of interest. And basically, they allow to have this really small field of view, but really high spatial resolution on the cortex. So basically, we position it really on top of in this case, of the middle temporal cortex. So right here on the right and on the left part of the patient. And with this, we were able to achieve both high spatial resolution of 1.2 millimeters isotropic and 800 milliseconds recording for the fMRI in humans. And then we acquired the standard anatomy. So this was the task. So we wanted to investigate a mechanism of speed encoding. So how we process visual motion in the brain. And we use different checkerboard moving for 1 second and then staying still, and with different components. So with different temporal spatial frequency, meaning that the size of the wedge were changing or the change in black and white were changing. So the final speed basically were changing. And we have like 5 combinations. In addition to that, since we have this really fast TR, we could do like the convolution and model for each voxel of the brain, model the response of each voxel, so of each population of neurons to this different feature of motion. And we know from physiology that there are 2 models that explain speed in the cortex. One is the separable model, so where you have the spatial and temporal. If you look at the spatial and temporal frequency component, and the speed is the ratio between the 2. In this case, the firing of neurons really depend on the spatial frequency, meaning that even if the speed is the same, the neurons is responding differently if the size of the stimuli is different. And in the other model instead is the speed tuning. So basically each neuron is responding always the same at the same speed, no matter which is the size of the stimuli. So what we did, so these are the map that is showing of the really on MT. So we also have an localizer of the middle temporal cortex. And for each of them, we applied the convolution to extract the HRF. We have this 800 milliseconds, so we have really nice shape of the HRF extracted. And we have this for each of the 5 conditions that we use. We extracted the maximum of this curve, meaning that for each voxel, we have the maximum response for each combination of spatial and temporal frequency. And we apply the model that I show you before. So some voxel will be more explained by this model, and some voxel will be more explained by the speed tuning model. So having the same speed for the different spatial and responding to the same speed, even if the spatial and temporal frequency are different. And what we actually show is that the region, the exact MT is mainly responding in a separable fashion. So really like the primary visual cortex. And we also find a specific response for spatial frequency within the complex. So we find the topography for low and high spatial frequency. And this is quite important because it was the first time it was demonstrated in humans that the secondary visual cortex is responding to a specific feature of the stimuli. And this also opened many questions about how we compute motion in other different modalities. Meaning that this area is really dependent only on the spatial and temporal frequency, no matter in which senses we perceive motion. Because if we perceive tactile motion, once again, we have a space and time combination, and auditory motion is the same. And this was really possible for the high spatial resolution and temporal resolution that we had with the 70. And this, open to the second question. So in the first part, so we really wanted to target an area and really trying to model the response of that area. So we wanted really to have a small field of view with high spatial and high temporal resolution. But when we go to more cognitive processes, usually we want to map the whole brain. And this, to allow whole brain fMRI at 70, we always need to find a trade-off once again. And I have to say that the most advantage that we have on 70 with the whole brain fMRI is the high signal-to-noise ratio, because this really allows us to have less number of trials to achieve significance, and we can apply some machine learning classifier that, like in 3 Tesla, is much difficult to achieve. Because like for an experiment at in 3 Tesla, you need like 5 hours. You will need 5 hours. You can do in 2 hours with the 70. So for human fMRI, this is something that really changes the field. And MVPA was more and more used on that. So these are other 2 works, 70 and investigating more like a whole brain approach, the multisensory brain. So as I said before, we always known the human primary visual cortex is devoted to visual information. So we prove actually how visual cortex is encoding motion from vision. But there are more and more studies, and also thanks to fMRI, there were more and more evidence that actually the visual cortex is multisensory in function, meaning that it's responding to auditory, visual, and tactile information. And this I will show you a first part of this work, really on a multisensory motion system at 70. And the second part more how multisensory process impact perception and behavior in real cortices. So the first part, the functional characterization of the multisensory motion system was done here at the 7 Tesla, the CIBM, and is part of the Marie Curie project that I was awarded recently. The main question of this work was really to see how neuronal population in the brain respond to different feature, to different sensory stimulation, and in particularly to motion via the different senses. The main principle is that what we usually know from literature is that we have a specific area responding to visual motion, a specific area responding to auditory motion, and a specific area responding to tactile motion. But there are more and more evidence that actually the brain is multisensory. So we have areas that are devoted to a specific task, so to motion instead of each sensory stimulation per se. And the goal of the project is really to see if we have neuronal population, the same neuronal population responding to the 3 sensory modalities, or if we have like different cluster in the same area responding to the 3 sensory modalities. So basically to find if there is a shared functional architecture within motion sensitive brain regions that is independent from sensory input modalities. And we actually did that using 70 scanning. This was the scanner here. We use a standard multi-band gradient API sequence. In this case, we have 1.5 millimeters and 1.5 seconds because we really want to map the whole brain and look at multisensory motion responses in the brain as a whole. And we also acquired the anatomical scan. These are the tasks that I use in the scanner. So I want to have exactly the same feature, but in the 3 different modalities. So the first one was visual motion. So the subject inside the scanner was looking at dots moving from right to left and left to right with 3 different features. So random dots, low spatial frequency and high spatial frequency motion. Then a second part was auditory motion, and this we did together with Leuven in Belgium. They have a device with speaker, and you can play the speaker from left to right and then from right to left. So basically, you have this sensation of motion going from the right here to the left here, and vice versa. And you can also decide how many speakers to play. So we play with that to achieve the same spatial frequency of the visual stimuli. So basically, we play only the first speaker, then we jump to and we play the third, and so on. And we did the same for the low spatial frequency. And the third one was the more challenging. So I had this magneto-compatible tactile device. So you can see me here. I'm really manually moving the wheel inside the scanner while the sequence was going on. And also together with Daniel Vance that designed the grid and the 3D lab here in a PFL, we print these 3 grids. And once again, we wanted to reproduce the random dots in the tactile domain, the high spatial frequency and low spatial frequency in the tactile domain. So we have these three modalities. As I said, thanks to the 70, we could have only 2 sessions with all these 3 modalities and have enough trial to perform both standard statistical analysis, and we are going now to perform multivoxel pattern analysis, so machine learning classifier, to try to identify in the brain which voxels are responding to the 3 modalities or only one of that and the difference across them. I'm going just to show you some preliminary results that I really did in the last weeks. So this is the surface reconstruction of the anatomy with on top the functional response for the standard GLM analysis. So this is for the visual motion task. So you can see that there is a really strong activity in visual cortex and the middle temporal complex. This is really as expected. And I was really surprised to see this is for the auditory motion task. So you see auditory cortex as expected. But we also find this strong cluster in visual cortex. So really proving the importance of visual cortex also on auditory motion information, not only on visual information. And I'm currently analyzing the tactile task to see if we have this hub in V3 that is present. Yes, less visible, but is here in all the 3 different modalities, and also performing more sophisticated analysis to see if we find cluster also in the middle temporal cortex for these three modalities. This was one application of whole brain 7 Tesla to multisensory, try to respond to multisensory information. And I'm now showing the last work. This work was done in collaboration with the Spinoza Center in Amsterdam and with Wiske van der Zwaag, that I think some of you know. She was here some years ago. In this work, we try to link multisensory processes also to perception and behavior. So we want really to see how audiovisual integration is done in the brain, and if combining together auditory information to visual information can enhance visual function. So can provide more information about visual function. So this was this is the task that we use. So we use illusory contour. Illusory contour is the ability that we have to perceive border even if no visual input are present. So in this case, like primary visual cortex, you don't have any information from this part of the visual field in primary visual cortex, but because there is nothing there. But we are still able to see the square. And this is something that is preserved even in animals. So really from across all the species. And since it was already proved behaviorally and also in literature that sound really help us to complete the image. So we are faster to see if there was a square here, if we combine a sound with the visual information. We wanted to see where actually in the brain this was happening, and if sound were really enhancing the response in visual cortices. So really helping in, giving a response of visual cortex where the border were missing. So in order to do that, we also use 2 kinds of analysis, because we want first, so these are the 2 kinds of analysis that we can use in fMRI. we wanted to first know where in the brain and how much in the brain we were computing this illusory contour process with and without sound. So this is a standard approach for which for each voxel we determine a response. So we can quantitatively say a response for each condition. So let's say for illusory contour without sound and illusory contour with sound. And we see the voxel in a unidimensional space. So we really respond about selectivity. So which area of the brain are responding more to a specific task. But thanks to the the high number of trials that we have, we can basically see the brain. Here is simplified in 3 voxels, but you can see it in like old voxel of the brain in a multidimensional space. In this case, for each condition, you have a dot in this space, meaning like for condition A, voxel 1 has less response compared to 2, and compared to 3, so we'll be here. And you have repetition of the same trial. So you have like 4 different responses for the same combination of voxel. And what you can really do, you can use linear classifier, as we use in machine learning techniques for different techniques, on fMRI data, so on ball data. And you can try to trace a line and basically find different brain states, so different part of the brain, where the information about, in this case, the illusory contour is processed. So here we are responding more about the representational information, or what kind of information is encoded and where. And this can really provide 2 different answers to the same question. And this is what we apply to this data. So we have like 17 subjects in the scanner, and they were seeing these 4 Pac-Man that you see here. And there were an illusory contour, so in this case a rectangle presented in one side of each part of the square. So it could appear here, 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 or here. And there were some trials where there was nothing, so completely no illusory contour. So we had some trial with no sound, so only to this, and the subject has to respond yes or no if you see the illusory contour or not inside the scanner. And then we have the same trials, but this time we added a burst, so a sound in the using headphone. And we had the same conditions, so IC and non-IC conditions. So we have these 4 conditions. And we did like a whole brain, 1.5 seconds and 1.5 millimeters whole brain fMRI scan. So what you can see here, if we look just at the GLM, so at the selectivity, we found the sound enhanced both response in the whole visual cortex. So it really increased the response in visual cortex. But this is not saying anything about where really sound is helping in recreating the illusory contour. So in having this enhancement of visual information, but we can see that we have this stronger response for the sound in terms of bold response when the sound was there. Then what we did this whole brain MVPA analysis with a searchlight approach. So like really for all the brain. And what we actually found is this shift in response. So basically when no sound was present, we found, we could decode the IC versus non-IC condition more on primary visual cortex, while when sound was there, we found this representational information more decoded in B3. So more in secondary visual area. And actually secondary visual area is where we create objects, and we have like object grouping and object formation. So if we basically if we subtract the 2, we were able really to see that we could decode more with sound in secondary visual cortex compared with the primary visual cortex. And this has a lot of also possible clinical application, because sound is really helping recreating the object in the secondary visual cortex, so in mid-level vision. And we can use this audiovisual integration also to try to restore visual function or to enhance visual function abilities in patients. And this is also something that we see also in the tactile domain. So for instance, patients with stroke now, they are in the treadmill and they are also looking at image moving, for instance. So you have this both tactile motion information and visual information combined together to enhance responses in the brain. So I hope I convince you that we can use 7 Tesla fMRI to explain spatial temporal neuronal computation in vivo. And we can also use to have a new insight into distributed neuronal representation. I provide an example with the illusory contour task. So I did the slide on what's next, what's the future now on 7T. And what I think is a field that now will go really on. So right now I show you that if you go really high spatial resolution, you lose in temporal resolution and vice versa. So it's really hard to obtain a combination between the two. But there are now more and more sequence development on this. There are new studies that show that we can go sub-millimeters, so really like 700 micrometers and go laminar, for instance, in primary visual cortex, and also go sub-second. So really go really fast with the fMRI. There was this paper in Science now, well, it was in mice, showing that we can really acquire data every 24 milliseconds with BOLD, even if still the spatial resolution was not that efficient. And what now is a lot used in clinical application that I also saw in Utrecht is the clinical fMRI mapping for epilepsy, the presurgical localization for tumor removal. So with 7 Tesla, we can really have topographical maps, really high spatial resolution maps of somatosensory cortex, language area, and different area. And the surgeon are starting to using it for before doing removal of epileptic tissue. So to see, like, to not remove language area or part of the brain. And also now there is a lot on machine learning applied to resting state, especially in Alzheimer disease, to see if we can use the fMRI in resting state to have information about the disorder, neurodegenerative disorder mainly. With this, I want to thank Micah and all the lab, the CIBM and Li Jing for all the scanner, and Dimitri and Pina. The biotech didn't show it here, but I'm also doing some TMS experiment on the motion part. The Belgian groups and all the people in the Netherlands, and the funding, obviously. Thanks for your attention.